Welcome to the IMAG MSM webinar series, and I am pleased to have the IMUBeam working group host this webinar, and I will pass it on to Steve.
Thank you, Grace. Okay, so today's webinar is going to be by Dr. Robert Latour, who's the McQueen-Quattlebaum Professor in the Department of Bioengineering at Clemson. Dr. Latour did his Ph.D. in bioengineering at the University of Pennsylvania in 1989, and then went immediately to a faculty position in bioengineering at Clemson, and I guess he's been there ever since. And he's fairly unique in that he does quite a lot of high-level experimental and modeling work at the molecular level, with a particular focus on the interface between proteins and surfaces for biomedical applications, but I think very, very widely applicable.
I'm fine. How are you?
So with that, I'll turn it over to Professor for his webinar.
Okay. All right. So, all ready to get started? All right. So, just introduce myself. Thank you, Steve, for the introduction. And as Steve mentioned, I have both a computational group and an experimental group, and this lets me do some unique things where we can really tightly collaborate between the two groups to synergize our interactions and the results or in the different methods we're using to complement each group. And the title of my talk is on the screen is Bioactivity is the Name of the Game, Multiscale Modeling to Predict Structure and Bioactivity of Adsorbed Proteins on Biomaterial Surfaces. So everybody can see the slide that just changed, I'm assuming. So I, as in bioengineering, very interested in the biocompatibility and biological response to materials put in the body, shown here in a hip joint or heart valves and a vascular stent. And of course, we're interested in how the body interacts with these materials when they're placed in the body. And one thing that kind of underlies things is that cells in our body don't have receptors for synthetic materials. And so inherently, they don't have any immediate way to respond directly to the material. So what happens when these materials are placed in the body, proteins in the body fluids rapidly adsorb on the surface, and they are what make the surface now bioactive. And so it's the orientation and the conformation that controls the bioactivity of this adsorbed layer of proteins. And this is really what drives the biological response to materials. So this has been a major area of research in the biomaterials field to try to understand protein adsorption and then how to design materials to control it, because this is the link to control biological response. And other than biocompatibility, the bioactive materials is a major theme in tissue engineering and regenerative medicine and many other aspects where we want to integrate bioactivity into our biomaterial system. And the way we do this is by typically using peptides and proteins. So for tissue engineering and regenerative medicine, we link biomolecules for cell signaling, drug delivery systems. Of course, we have cell targeting and intracellular delivery mechanisms. Bioimaging is tissue targeting, where we link bioactive molecules to target the imaging system. And for enzyme-based biotechnology, of course, for bioassays, biosensors, and bioreactors. So for all of these applications, the real key issue is maintaining the bioactivity of the peptides and proteins that are being used to perform the various bioactive functions. And this requires the accessibility and the proper conformation of the bioactive sites of the proteins that are presented. And just to sort of illustrate this with some cartoons, if we look at an enzyme in solution, of course, it has a bioactive site. And when that adsorbs, if it absorbs in a proper way, we can retain the bioactivity, and it can perform its function where it's binding to a substrate and causing some sort of desired reaction for a biosensor, for example. However, if the enzyme is adsorbed in the wrong orientation, the active site can be blocked sterically and therefore making it inactive, or if it's conformationally distorted when it adsorbs, you can lose bioactivity that way as well. So we'd like to understand these processes to be able to control the bioactive state of our system. And this requires really atomic level understanding and extension then to higher length scales, so we can understand what these peptides and proteins are doing on our surface and how they're interacting with the cells. Experimental systems are largely, or experimental design currently is really relegated to a trial and error black box approach, because we really don't understand things from an experimental approach at the atomistic level that we need to really understand what's going on in our system. So, for example, often you'll see cartoons like this in papers where people are linking, for example, antibodies or other bioactive molecules, proteins to a surface, and they have them idealized, all standing upright and nicely organized. But in reality, it's much more likely that we don't have the kind of control we want of this orientation, and we lose a lot of bioactivity if we don't have the conformation and the orientation control. And this is where molecular simulation comes in, because this provides the ability to visualize, understand, and predict these kind of interactions, both from the atomistic level all the way up to the macromolecular length scales through multiscale modeling. And because of the size of these type systems, the empirical force field-based simulations are the main type of molecular simulation that fits this type of problem, and both all atom and then coarse-grained multiscale approaches. And just to have an example of how the surface can affect bioactivity, here's some results from some simulations that we've done before. This is an LK peptide, leucine and lysine. Leucine, of course, is hydrophobic, nonpolar side chain, and the lysine is a positively charged side chain. And these peptides in this sequence form an alpha helix on surfaces. And here I'm showing a carboxylic acid surface, and just showing the simulation predicts that this will adsorb with the positively charged lysines adsorb down on the negatively charged surface, with nonpolar leucine facing towards solution, and it maintains its alpha helicity on the surface. And if we look at a different surface, here's a hydrophobic surface, and this shows a quite different response. Here, the nonpolar leucine amino acids are adsorbed down, and the positively charged lysines are up, and we've lost the helical structure. And so this just shows an example then of how on our surface we may have one situation where we're retaining bioactivity, and another where the bioactivity is lost simply by the way the protein is interacting with the surface. We need molecular modeling really to get down to this level to understand what's happening at the atomic scale. So with that background, the objectives of my presentation are first to provide an overview of the principles and the methods that we use for molecular simulation of protein-surface interactions, and then to introduce some of the challenges that we face in transcending time and length scales. And we've been spending lots of effort over the past decade understanding these problems and addressing them. And one approach is using advanced sampling and to handle a time domain problem, and then multi-scale modeling to handle length scale issues. And our present approach to develop these capabilities are first to develop an interfacial force field. These are the parameters that control the interaction between the proteins and the material surface. And presently we've been focusing on all atom methods for this, and we're planning on extending this to the coarse grain level to extend our length scales. And then we've worked on program modification to let us use these interfacial force field parameters. Most of your molecular simulation programs are developed to just use one force field for the whole system. and the interface is just falls out of the parameters you're using for the solution phase versus the solid phase. And we found, and as I'll show, that we really need a separate set of parameters to treat the interface as a separate phase in the material to be able to simulate the interactions between proteins and surfaces correctly. And then once we have developed interfacial force field parameters to accurately represent how the amino acids of a protein will interact with surfaces, we'll then can apply those to simulate protein surface interactions. And then we can we're looking at extending that to higher time and length scales through coarse-grained methods. So here's a depiction of a protein. This is a segment of fibronectin. It's color-coded by amino acid type to just show the high amphilicity the proteins have, where each amino acid has a different chemical character and adsorption character. And the way the simulations are run is that you have a series of parameters that define how the atoms in the system interact, and these are represented by bonded interactions, and then electrostatic interactions, and van der Waals interactions that then determine how the amino acids of the protein will interact with functional groups of a surface. And we also, what's very important in the system is the solvation effects, both of water and the ions. They're also very dynamic interacting elements of the system that we have to make sure we're capturing as well. And for example, here the RGD is the bioactive segment of fibronectin that mediates cell receptor binding through integrin binding. And if we're going to simulate the protein and keep it bioactive, we need to be able to adsorb it to the surface with the RGD site solvent accessible and conformationally intact, so it can bind to the ligands of the cell receptors and make this bioactive on our system. So first I wanted to go through, for those of you who might not have much background in molecular simulation, a little bit to explain what the force field is about. And for empirical force field-based simulations, we have an equation that represents the potential energy of the interactions in your system. And this is divided into several different terms. So the first term here is shown for bond stretching for covalent bonds. And then we have terms that handle covalent bond bending for 3 atoms that are covalently linked. And then another term for bond rotation, the potential energy related to the torsional rotations of a bonded chain. And we have non-bonded parameters using Coulomb's law. Each atom is designated with a partial charge, and Coulomb's law then predicts how these charges interact. And then there's a Lennard-Jones 12-6 potential is used to represent the interactions for van der Waals interactions in our system. The key parameter here being epsilon, which is the well depth of the interaction between your atoms. Now, the real power in this system, this method, is that if these parameters are all properly representing how our atoms interact with each other, so for example, here in the bonded term, we have K sub B is the bond stiffness, and B sub O is the equilibrium bond distance, and the same type of parameters for each of our different components of our system. If these parameters are all set to represent properly how the atoms interact, you can apply this for both all atom and also coarse-grained modeling to scale up your system to a very large degree, and all the atoms in the system should be interacting with each other appropriately. The downside of this method is that your parameters are not set right. You can still run your simulation, but the results will have nothing to do with reality. And so this is a key parameter in these force fields, is to make sure you have the force field parameters set properly to represent your system accurately. And this is an area that we've really sort of pioneered the field with, of developing interfacial force field parameters to make sure we're getting how the amino acids interact with surface functional groups properly, so we can represent protein adsorption in a realistic manner. Now, the reason why this is called a force field equation is because when the energy equation is differentiated with respect to position, it gives you the forces acting on the atoms. And then using Newton's law, we can get the accelerations and therefore do molecular dynamics of how atoms move over time. So next I'll go through some of the basic equations for molecular dynamics. We'll start with initial positions and velocities, which is a function of the temperature of your system. And once we have these initial positions set, then we plug them into our force field equation to get the forces acting on our atoms. Then that's the forces are divided by the masses, which give you the accelerations of the atoms. That's integrated over a time step, typically 2 femtoseconds, to follow the atomic motion, gives you new velocities of your system. That's integrated over your time step to give you new positions of your atoms. So that's one cycle of your MD simulation. If we go through 10 to the 6 or a million cycles, we have 2 nanoseconds simulated, which is obviously an enormously short amount of time that has nothing to do with our experimental measurements. And this is a big issue where we've got to transcend these time limitations to get simulations to provide results that are comparable with experimental methods. And then using statistical mechanics equations, we calculate the temperature and the pressure of our system, which is a function of the atomic position and the motions and the forces between your atoms. Okay, so next I want to address the challenge of how we do our simulations in a way that can match what we're experimentally trying to compare them with. And there's a big challenge in the equilibration of our system. So if we look at the difference here, for our experimental measurements, we have average behavior over billions of proteins in our experimental system, and our time frames are typically seconds, hours. Compare this to a conventional molecular dynamics simulation. We're looking at the average behavior of a single protein, for example, in our simulated system with time frames of tens of nanoseconds. And obviously there's an enormous gap between these two ranges of the experiment compared to our simulation, and we've got to find a way to bridge this gap. And the way we do this is we use some clever ways with our simulation. First, we use periodic boundary conditions to represent a unit cell of a unit system. That lets us have one small system that can represent really an infinite size, just like a unit cell in a material crystal. But then we also use advanced sampling methods that lets us transcend the time scale, and I'll say a few things about this, where we use statistical mechanics algorithms to help us bridge our time scale and represent an equilibrated system as opposed to a time scale based system. And then coarse-grained methods let us extend the length scale by grouping atoms together and reducing the degrees of freedom in our system so we can greatly enlarge the size of the system that we're representing. So first, to say a few words about some of the advanced simulation methods, the accelerated sampling methods. One of the most popular ones is called replica exchange, in this case temperature replica exchange. And this plot just diagrams out what we do with replica exchange. So on the top row here, I'm representing what we have with a conventional molecular dynamics simulation. We just representing two different energy wells, the barrier in between them. And in conventional molecular dynamics, you're often starting in a non-low energy part of your system. It's often trapped there. We can run the whole simulation and result with a resulting set of states that have nothing to do with the real equilibrated system. And that's because it's called energy well trapping. So with replica exchange, we solve this by running multiple replicas of our system parallel. We run typically 24 or up to 42 replicas. The baseline replicas at our body temperature, for example, and then the other replicas are all simulated at exponentially increasing temperatures. And then this lets us run our simulation, and with the excuse me, with the pardon me, with the higher temperature replicas, the temperature lets us jump out of these energy wells and escape from them and explore other regions of the phase space. And then there's an exchange process that's done between their temperature levels that lets us exchange when a higher temperature falls into a lower energy state. It's exchanged down the temperature ladder. And the result of this is we end up with a Boltzmann-weighted distribution of states, which should represent the equilibrated state of our system, and this can be directly compared with experimental results. So by this, effectively get away from our time restraints and let us get an equilibrated system that should be comparable to our experiment, assuming our experiment is also represents an equilibrated state. And we've done a lot of work with a new version of this called Tiger 2, which represents temperature intervals with a global exchange of replicas. It's particularly a more efficient replica exchange method that we particularly are designing for big systems like proteins absorbing on surfaces. And then to transcend length scales, the main technique is coarse-grained modeling, and showing an example here. In a coarse graining, you take groups of atoms and you represent them by individual beads in your system, and then you set the force field parameters between your beads to represent the behavior of the groups of atoms contained within the beads. And then you also couple this with an implicit solvent group or expression in your force field, and this lets us get rid of having to represent water as an individual molecule in our system. And this greatly reduces the degrees of freedom and lets us greatly scale up by orders of magnitude of the size that we can run in a simulation. And so with a coarse-grained method, you then run your simulation and equilibrate it using your advanced sampling techniques. And then after you're equilibrated, you can reverse map it back to an all-atom depiction of your system and do some further equilibration to get an equilibrated system for a much larger system than you can handle with straightforward all-atom molecular simulation. Okay, so next I'm going to transition to show some of our results where we've been working on tuning the interfacial force field that we need for accurately simulating how proteins interact with surfaces. And this is work that we've been conducting essentially really over the past 10 years to refine these methods and apply them. And so we're focusing on the amino acid surface interactions as really the fundamental unit in our system. And we use adsorption free energy as the key parameter that really represents how the binding affinity of amino acids for different types of functional groups on the surface. And experimentally, we measure Gibbs free energy of adsorption through adsorption isotherm measurements. And this is gives us the Gibbs free energy. We do this under constant temperature and pressure. In our simulation, we actually are calculating Helmholtz free energy, and we develop methods to do this. This is under constant temperature and volume, but in the condensed phase system, they're effectively equivalent measures because we have very little change in volume, the adsorption process. In our experimental methods, we use surface plasma and resonance spectroscopy to measure the adsorption isotherm to characterize the binding of peptides to our surface, and we use alkane thiol SAMs for model surfaces, and then also we spin copolymers to do polymeric thin films. But there's many materials that we can't use with SPR because you need really nanometer thick films for this, and we've developed a method using linking this also with atomic force microscopy for these other type of surfaces as another way of obtaining measurements of free energy of our system. And then for our molecular simulation, we can calculate adsorption free energy so we can compare with experimental data. And this is one of the big limitations here, that there's very little experimental data that can be used for comparing with your simulation results. And I think our group is really really led the way in the field to try to develop these methods to get this kind of data. And once you have this data and you can calculate it with your initial force field set, then you can compare your simulations with your experiment, and that tells you where you have to adjust the force field parameters to correct errors so you can properly represent the protein absorption behavior. So I'll go into our experimental methods now for how we've measured free energy of absorption. Up in the upper left, we use a host guest peptide model where T and G are threonine and glycine, and they provide solubility and extra mass for our peptide to be detected by SPR, surface plasmon resonance. And then the middle amino acid X is varied over all our different amino acid types, which changes the absorption character of the peptide. Then we have our surfaces, either using alkane thiol cell assembled monolayers or spin-coated polymers. And in the bottom, we show just a schematic of surface plasmon resonance. And surface plasmon resonance is a technique where you measure the mass absorbed in real time, and it's very effective to get absorption isotherms, where you do your absorption experiments over a range of concentrations, and you measure the equilibrated amount of protein absorbed under each concentration. And from this, we get, here's a typical kind of data plots where we look at the amount absorbed under different solution concentrations. We plot the response, and we work out the equations then to give us the free energy of absorption for each of our peptides using this method. And then for the different material, to expand our ability to use materials that aren't really conducive for SPR, we did a large set of studies with alkane thiols, which we can do with AFM and SPR. And by linking our peptide to our AFM tip, we can measure the debonding force from our surface. And on the upper right, we show the correlation between our debonding force from our AFM experiments and our free energy measured by SPR, the same type of peptide surface systems, and showing we get very nice correlation between the two. And so we use this then for surfaces like silica glass that can't be readily used with our SPR, but we can use this with our AFM, measure the debonding force, and get an associated effective free energy of the interaction of our peptides with that surface. And we've done this for silica glass, polymethyl methacrylate, and polyethylene. And this plot just shows all the data we've generated over the several years. We have about 14 different amino acids we've looked at over a wide range of surface chemistries, and we've gotten the free energy of absorption table that provides our benchmark experimental data that tells us this is how the peptides are supposed to be absorbing on these surfaces. And this now provides the data set for us to compare with for our simulations to see if the simulation, the force field used during the simulations is providing us with reasonable results. And from our AFM, we also then show the values for our polyethylene, polymethyl methacrylate, and our glass. So now we have our experimental data set that tells us how these peptides are supposed to be absorbing on our surfaces. And now we can go to the simulations to see how that compares. So here's an example of our simulation system. Again, we're simulating the same host guest peptides over a material surface. And we're using the CHARM program and the CHARM force field, which is one of the major protein force fields that are used. And we're going to calculate the absorption free energy to compare with our experiment. And at Clemson, we use the Palmetto cluster, which has over 20,000 processors, all in a big computer farm. It's an excellent resource for research computing. It's fully staffed and monitored 24/7. So this has been a great asset for our simulations. On the right, we show a typical type of model system. We've got, this is showing a alkane thiol cell assembled monolayer surface with our peptide in explicit water over the surface. And in our simulation, what we do is we monitor the position of the peptide over time as it's absorbing and desorbing from the surface. which gives us a probability distribution of the peptide over the surface. And it's very analogous to the concentration profile that we get experimentally, and this lets us calculate the absorption-free energy based upon the probability distribution of peptide over the surface. And if we just did a conventional molecular dynamics simulation with this system, the peptide would strongly absorb to the surface and not come off for the whole simulation. And this would mean we wouldn't be able to calculate free energy because we need the probability distribution of the protein or the peptide in all positions adsorbed to the surface and free above the surface. And so to overcome this, we use an advanced sampling technique called umbrella sampling. And this slide just shows an example of that, where we use a biasing, a restraining potential to just sample the peptide in certain regions, and then we change that region incrementally from the close to the surface all the way to being far above. And then we can link all the probability distributions we get from this and put this all together, and this can let us get the probability distribution we need for calculating free energy. And so this just shows an example of the peptide restrained 24 angstroms above the surface in terms of the center of gravity of the peptide. The dots you see here are all the water molecules we have fully represented in the system, and we will simulate the system. Is there a question? Okay. All right. And
So go ahead with your question if you wish. Jacob, you had a question?
Yeah, I did have a question. It's about the lengths of those simulations. So usually how big are they? So you said you use a 24,000 nodes cluster, but actually how many nodes for how long do you use, and how many of those molecules or atoms or entities are you simulating? Millions at a time, thousands at a time, for how long?
Okay. These simulations that I'm showing here are about 15,000 atoms total, or I'm sorry, more about like 12,000 atoms total, mostly made up of the water, which are individual water molecules, and we'll simulate this in parallel with 8 processors per node, simulating this for about 25 nanoseconds, which will typically take a few weeks of simulation to get the result.
2 weeks in simulation for 8 cores, 8 nodes?
We'll typically use 1 node, and we'll use 8 CPUs on the node, or 8 cores on the node. But we'll be running 24 of these in parallel, because we'll have them all separated by 1 angstrom separation, and we'll run 24 in parallel all at the same time. So we'll be running with 24 nodes, each with 8 cores per node.
Thank you.
Okay. And then on the right just shows the same type of umbrella sampling, showing the other extreme down at 4 angstroms separation from the surface, where you can see the peptides clearly interacting with the surface. And we'll do this for, as I say, the peptides separated by 1 angstrom increments at every position over the surface, and this lets us put this all together into one single probability distribution that then we integrate over and use this called a probability ratio method to calculate the absorption free energy. So I'll show some results here for one of our recent ones. There's a silica glass surface in our host gas peptide over the surface, and we started off using charm parameters as our default parameter set. So we charm parameters for the peptide in solution, and there were published charm parameters for water interacting with the silica, and we were just assuming that we could use the charm parameters for the protein in solution or the peptide interacting with the silica based upon the existing parameter set. And then we have our experimental benchmark data set of our free energies to compare this to see how well the simulated results using the default parameters are giving how well they're representing our free energy. And I'd like to add here that you'll see a lot of groups doing publications where they're just using this type of simulation and really not paying attention to what's going on at the interface and just assuming everything is fine. And as I'll show you, our results show that everything is very much not fine. And if you just run simulations this way, you'll get results that probably have very little to do with the real system. So here's the results. Here I'm plotting the simulation results for our host gas peptide with these 8 different amino acids against our experimentally measured values. If they all lined up perfectly, all the data would fall along this dark black line. The dotted red line represents 1 kcal per mole energy difference from the target value. And as you can see, the charm parameters greatly overestimate the binding affinity of all our amino acids. They're all about 8 kcals per mole too high, 6 to 8 kcals too high in their binding affinity, which is, if you ran this then as to simulate your protein would absorb much too strongly to your surface and not represent anything like the experimental method. So this is where we've really been a pioneering group demonstrating that you can't just take these existing parameters and assume they're going to work for your interface, because the interface has a molecular environment that's very different than either the solution phase or the solid phase itself, and has to be treated as a separate phase. So the problem is we can't just go in now and change the parameters of our peptide to get it to absorb properly to the surface, because the parameters we're using are designed to control the conformation of the peptide in solution. And that's just as important if we're going to represent protein absorption behavior. We have to get the conformation of the protein in solution and the interactions of the protein with the surface, both of those accurately represented. The charm parameters are tuned to get the peptide conformation correct, and what we're finding is they're not representing interaction with the surface correctly. We can't change those parameters or we'll get the conformation behavior wrong. So our approach to solving this problem is to introduce this interfacial force field and modify the charm program to let us use it as an independent force field that's controlling whenever an atom of the solution phase is interacting with an atom of the surface phase. It'll use this separate set of non-bonded parameters to control those interactions, while we're still using the regular charm parameters to handle the peptide behavior in solution. And we can use a material science force field to handle how the surface itself is behaving. And this gives us an independent way of tuning in the adsorption behavior without messing up the conformational behavior, which should be handled by the regular charm parameters. So for this, this shows the results of our tuning process, where we go in and we incrementally change. We found the Leonard-Jones parameters. The well depth is the most important parameter, and by weakening those parameters for how the peptides are absorbing, we were able to pull down the adsorption affinities to line up all within a half a kcal per mole, well within that, to match our experimental values. So now this lets us excuse me, this lets us tune our system so our amino acids should be interacting properly with our surface. And with this capability, then we can now have confidence to apply this to a protein absorption process and represent properly how the protein's absorbing with our surface. And we've done this. Here are the results with polyethylene. Again, showing the simulation results on the y-axis versus the experimental results. If you look at the red dots, those are the default charm parameters, showing there's actually no correlation between the amino acid, between the energy results from the simulation versus the experiment. And by tuning these parameters, we've brought them all well within close to a half kcal per mole energy compared to the experiment, and greatly enhanced the correlation between these two. So this is the interfacial parameter set for peptide amino acids interacting with a polyethylene hydrophobic surface. And we're also just finishing up a set with polymethyl methacrylate as an amorphous polymer surface. And again, here you can see the blue diamonds are the default charm parameters, and there's actually no effect of none of the amino acids really absorb any differently than each other, whereas experimentally we get very large changes, and we're able to tune these parameters again to bring the interfacial force field tune parameter set such that we're getting the amino acids interacting with our surface much more closely aligned with our experimental data set. So now that we have the ability to simulate amino acids interacting with surfaces, we can extend this up for simulating protein absorption behavior. So that's the next part of my talk. And so now we have interfacial parameters that should interact between the amino acids and our surface. First, here's silica glass. And now we can simulate the absorption behavior of a whole protein on the surface. And we should be able to accurately simulate how the protein's absorbing and unfolding on the surface. Excuse me. And these are now much larger systems where we have 20,000 water molecules and about 2,000 atoms in our protein. Now, the other part of this, then, we have to have experimental data that tells us if our simulation results are accurate or not. And there's very limited data out there that can be used at this level to tell us what the protein structure is on the surface, and we need this data to be able to evaluate our simulations. And so my experimental group has spent a great deal of effort developing these methods. And so the experimental methods here, we're using circular dichroism spectropolarimetry to look at the secondary structure of the protein on the surface. And then a method we've been working a lot with recently, amino acid specific labeling and mass spectrometry. This lets us probe the orientation and changes in tertiary structure of our adsorbed protein. And then we use standard bioactivity assays to look at the effective absorption of the bioactivity. And these are all things that we can compare with our simulation results, so we can assess whether their simulation results are reasonable or not. So for our simulation, we have our tuned interfacial force field, and we are applying our Tiger 2 advanced sampling algorithm to let us equilibrate the protein on the surface, because it's no good just doing conventional molecular dynamics. You'll just represent the behavior of one protein, and we need to represent the behavior of a multitude of proteins as an equilibrated system of structures on our surface. So just to say a few words about circular dichroism, I'm not familiar with it. You pass circularly polarized light through your sample, and proteins are chiral molecules. And if you look at wavelengths from 190 to 250 nanometers of your polarized light, passing left versus right polarized light through your sample, the alpha helices will give you a very different spectrum than your beta sheet or your random coil. And by doing this, we're able to look at the structural composition, secondary structural composition of proteins, both in solution and then after we absorb the protein to our surfaces, to see how the absorption affects the secondary structure. And again, this is data that we can compare directly with our simulation results. And for our amino acid labeling mass spec, we borrowed this from proteomics field, and it's based on the chemical modification of targeted amino acids, where we can apply chemical labels to the amino acid without disturbing the structure of the protein, and we can then assess how changes in absorption changes the solvent accessibility of these amino acids. So here's just a diagram showing the blue is labeled amino acids. That means they're solvent accessible, and the red's indicating buried amino acids that in solution they don't get labeled, and mass spec lets us identify which are labeled and which are not. Then we absorb the protein and do our labeling process, and this is just depicting, for example, if these bottom two amino acids show that they're unlabeled after we absorbed, that tells us that the surface is blocking the solvent accessibility of these proteins and giving us a clue about the orientation of the protein on the surface. Alternatively, if amino acids are not solvent accessible in solution, but after we absorb it, they become solvent accessible, this gives us a key about the tertiary unfolding of our protein, where the buried amino acids are now becoming solvent exposed due to the unfolding of the tertiary structure. And so we're using these experimental methods to get the kind of amino acid level data we need for assessing simulation results. And this just shows some of our CD results for lysozyme on silica glass. The axis here, y-axis, is the percent helicity. The native state is about 38% helical structure of this protein. And as you can see, we absorb it from different solution concentrations. We get very different changes in the amount of unfolding. At high solution concentration, shown the top blue line, you get a lot of protein absorbing very quickly, and the protein-protein interactions prevent the unfolding. And as you're absorbing it from more dilute solutions, you can see the proteins have more time to spread out on the surface before they bump into neighbors. And this is the and by doing this, we're able to get data that basically shows what the individual protein should be doing when they're absorbing on a surface, because our simulations actually will represent only a single protein on the surface. And this provides the data that tells us that what our protein should be doing on this type of glass surface. And then this slide presents some bioactivity data and then some of our early amino acid labeling data. And here we had lysozyme again on different alkane thiol surfaces. And we'll look at the methyl surface versus the OH surface, where we see very big differences in the bioactivity of the absorbed protein, absorbed lysozyme enzyme. And the question is this due to the conformational changes because the protein's pulled apart on the surface and unfolding, or is it due to the orientation of the protein where it's absorbed with the bioactive site down and not being accessible? And so the question is, what is really inducing these changes in bioactivity? And on the right here shows our amino acid labeling results. The top right plot, the amino acids shown in green are the tryptophans that line the bioactive pocket of lysozyme. And this is showing that they're all solvent accessible when it's absorbed on the OH SAM surface. So this site is available for the substrate and enhanced bioactivity. And on the bottom right, we see that these are all in gray, which mean that they're not solvent accessible and not labeled after this is absorbed on the methyl surface. And this tells us this active site is being blocked when it's absorbed on the methyl surface. And that's telling us that the conformation, the orientation of the protein on the surface is really what's causing this shift in bioactivity. It's not so much the conformation, but it's being on the methyl surface. The hydrophobic binding site is absorbing down on the surface and blocking the bioactivity. So just to wrap up now, now that we have our experimental methods developed so we can look at, have experimental data on how the protein is supposed to be absorbing on the surface, now we can apply our interfacial force field to simulations of the protein on the surface and use our sampling methods to transcend our time frame to get an equilibrated system of states of that protein on the surface after it absorbs. And then we'll be able to compare the structure of the protein and the solvent accessibility of the amino acids and assess the bioactive site to give us information of how accurately our simulation is representing our experimental data to validate, again, our interfacial force field that we tuned for the system. And these simulations are actually ongoing right now. Future plans, we plan to extend our time and length scales to be able to predict the behavior of a macromolecular system where we have multiple proteins on our surface. So the first step in this is and for this we need our coarse-grained methods. It's too big for all atom modeling with explicit water for this type of system. So we're going to be using coarse graining to get interfacial force field parameters, very analogous that we got for the all atom models, but we'll be developing it for our coarse-grained models of the protein. Then we'll be able to do our coarse-grained simulations of competitive protein adsorption behavior, which refer to as Vroman effects, where we'll be able to simulate multiple proteins in solution and even different types of proteins and look at how they compete for the surface and compete for binding with each other to predict the final adsorbed state of the protein layer on the surface. And this would really revolutionize our abilities if we were able to develop these capabilities, because this is not possible at this time. And then we're looking at future studies to extend this, where we can use our coarse-grained modeling of the known structure of some of the cell receptors, integrins, and how they're binding to the adsorbed protein layer. And again, then to assess how the adsorption process influences the bioactivity of our surface. And then we want to be able to extend this for surface design to be able to design in bioactivity in a much more knowledge-based method. So just to conclude then, we said a knowledge-based design of biofunctionalized materials requires atomistic level understanding of the bioactive segments and extension of this over multiple time length scales to predict macromolecular behavior of our proteins on our surfaces. These bioactive segments must be accessible to molecular targets to retain bioactivity, and they must be conformationally intact. And these are the key things that really are most of the experimental studies don't provide this kind of information, but we're designing our experimental systems to be able to assess this. Then through multiscale molecular modeling and simulation, we should be able to provide a powerful approach to predict and understand how these bioactive elements are integrated and interact with our biomaterial systems to provide bioactivity as we need to perform the different functions that we're interested in. And this serves as a potential to provide a very valuable tool for service design and optimization of bioactive biomaterials for many different applications, from the atomistic to the macromolecular levels. So I'd like to acknowledge on the left hand is my research group, both experimentally and simulation, really over the past 10 years, essentially. In yellow are the current group. Funding and computational support from NSF and the New Jersey Center for Biomaterials, ResBio, through NIH, and also NIH grants that we've gotten, and currently working on a DITRA grant for defense, and also our computational resources. And then several collaborators, particularly Steve Stewart in the chemistry department at Clemson here, is a great asset for our modeling, and various other investigators around the country. So with that, I'll be happy to address any questions. Thank you.
Great, thank you, Bob, for a terrific talk.
You're welcome.
So the floor for questions.
I can start with a question. So Bob, the one thing that was really impressive is the tremendous amount of validation work. So I was wondering if you could expand on that future direction slide. What are some of the particular questions you're thinking of tackling as you go to complex multi-protein systems?
Let's see, you're breaking up a bit. I'm sorry, could you ask that again?
Sure. So I was saying that there's a tremendous amount of validation work that you showed. So the foundation is really there to ask some larger, say more complicated questions that you alluded to in the future direction slide. So I was wondering if you can go a little bit deeper into that and talk about maybe some of the questions you're going to go after first in these multi-protein systems.
Okay. Well, really, the key issue right now is methods aren't in place to accurately represent these kind of systems. So the first way forward is to develop our coarse-grained models, like I showed with the all-atom models, to get the coarse-grained parameters and validate them that they work for protein adsorption behavior. We're starting with individual proteins, comparing with our experimental data. And then once we have those parameters, where we're confident that it's realistically representing what proteins are doing on our surface, then the next stage is to scale that up to show that we can simulate the competitive behavior. We have multiple different types of proteins in solution, how they compete for that surface, to end up with what kind of proteins are on the surface and what their structure will be in their bioactive state. That's one primary direction, and it's largely method development to show that we can accurately represent this type of a system and provide this then as methods for the community to follow up on for a broad range of individual applications. Another area we're extending this to then is when you're linking protein peptides into hydrogels, for example, to make bioactive systems, and we're looking at applying these same kind of methods to be able to predict how accessible these bioactive segments are on these other type of material systems that are being widely developed for tissue engineering and regenerative medicine. And there's another big area, of course, with drug delivery, where people were putting antibodies as targeting molecules and drug molecules into synthetic surfaces, nanoparticles. And this is another important area where the conformation and the absorption behavior of these bioactive molecules on these substrates is going to be critically important for how effective they are. And we'll look at extending those in where we can simulate nanoparticles with the antibodies, for example, linked to them, and look at that as a tool for design strategy, how you want to place your antibodies so they're maximally effective on your surface.
Terrific, great. I got a text from Guy asking to be unmuted. I guess he's unable to unmute himself. Can we unmute the viewers?
Muted, Guy.
I'm unmuted now?
Yep, can hear you.
Oh, great. Very brave of you. I really like the general approach that you have for looking at surfaces. Now, some of the stickiest proteins I know are also some of the longest and most disordered. Could you say a few words about the issue of sort of proper sampling? Are your methods going to be extendable to disordered proteins, like, say, amyloid beta or something, a fairly long but disordered protein, where you have to really sample a lot of configurations to be able to get to a minimum?
Yes, and I don't think that's really a special issue for your disordered proteins, because the thing is, when these proteins adsorb, as I showed for the lysozyme, which is a small protein absorbing, it was largely unfolding, where the helicity went from 38% down to less than 10%, and it's becoming largely disordered on the surface. So we're facing those issues right now, even with all our proteins. So I think the methods we're developing are specifically to let us get this equilibrated ensemble of states as opposed to a time sequence of the absorption. And these methods, we're starting with small proteins, and with our coarse graining, we should be able to go up to much larger proteins, including the amyloids like you referred to. And the whole idea of these advanced sampling methods are to rapidly sample a very large number of configurations, and using statistical mechanics to sort them all out to come up with an equilibrated set of an equilibrated ensemble of states that should be very representative of your equilibrated experimental system.
Very good.
I think Jacob had a question.
Yeah, it's continuation. I'm trying to figure out the computations that you're doing. So I figured out the capacity, and now I'm trying to figure out the techniques. One thing you said is like you are able to parallelize some things. So you have 24 things running in parallel. But can you parallelize more? So what kind of techniques typically do you use? Do you use finite differences, finite elements, Monte Carlo? What kind of simulations do you have? Trying to figure out what goes on there.
Right. Okay. We're not doing finite element. I think that could be an area we could I've done finite element modeling before on other solid systems, but right now we're using molecular simulation packages that are all force field based that predict how atoms interact with each other. And in fact, we're moving from charm to lamps. And I just spent the year before last a year sabbatical at King's College in London, working with an expert in the LAMPS program. LAMP stands for Large Atomic Molecular Massively Parallel Simulator. It's from Sandia. It's an open source, freely available code that's been widely used, and mostly it's developed for material science, but we're building in some of the missing parts of the biological system so you can simulate proteins properly. Now with LAMPS, you can scale up to hundreds of processors in parallel for the same simulation. In CHARM, it's very limited. You can scale up to about 8 processors, and then the communications start bogging everything down. So we're very excited about moving over to LAMPS, we just got a 5 million hour award from Exceed, which is an NSF computational program using the University of Texas Austin system. And we have planned simulations where for our protein adsorption on our silica surface, where we'll be doing 24 replicas, each replica with about 64 parallel processors running together. So all in all, we're talking about thousands of processors all on the same system, which is of course what you need for these kind of large complex systems if you're going to sample it within your reasonable time frame.
Oh, I think Guy is muted. Guy, you're muted again.
Guy, did you have a question?
I do have one more quick question. And first I should point out that Marcus apologizes. He had to run off right at 4 at your time. But the question Marcus would have asked is something that's of interest to him. It's something that you probably have a better feel for than anyone else, which is how far can a protein feel? What rules of thumb can you give for how far down into a crystalline solid or a surface can a protein feel?
It's actually quite limited. I'll answer 2 ways: how much the protein feels the surface as it moves away from the surface, and there once an amino acid is about 15 angstroms away from the surface, the water really blunts any of the interaction, shields the interaction. So when we look at our probability distribution, where we get a free energy profile of the peptide on the surface, once it's further away than about 15 angstrom from the surface, it's just like bulk solution. And so the protein, so we're talking about those kind of scales. So when the protein's on the surface, it's going to be feeling approximately the same, about 15 angstroms down into the surface. And any layer thicker than that, you really don't have to represent to capture a full dense system.
Thanks very much.
Great. So do we have any more questions?
And I'll just add one thing to that, Guy. That's the one really nice thing about the molecular simulations. I mean, these are questions that, simulation, I didn't know the answer of that until we started doing our simulations. Then you watch it, you go, wow, look, these kind of things are happening, and you can see these kind of events and really get down to the atomic level and start understanding and thinking at that level and appreciating a lot more what's going on.
Thank you.
Yeah.
Well, thank you. There's no more questions. I think we can end the webinar there. So thanks a lot, Bob.
All right. Well, thank you. Thank you all very much for attending. Appreciate it. Happy Thanksgiving to everybody.
Happy Thanksgiving. Bye.
Thank you. All right. Bye-bye.
Bye, Grace.